I'd like to welcome everybody to today's Connect Science lecture. I'm Tim Rebick from Dana-Farber, and I want to, before we get started, remind you that as you have questions for the speaker today, please put them in the Q&A, not in the chat. We'll monitor those and ask them of the speaker by the time the lecture is completed. So please ask those and we'll remind you of that. And the second thing is, please note the Connect Science series. You can see on the web here is on the slides here are the dfhcc.harvard.edu/Connect Science website. That site contains recordings of all prior lectures as well as a list of the upcoming talks. And so we hope that you can all participate in those. So for today, I'd like to welcome our speaker, Dr. Brian Wolpin, who from the Dana-Farber Cancer Institute. Brian is an associate professor of medicine at Harvard Medical School and the Robert T. and Judith B. Hale Chair in Pancreatic Cancer at the Dana-Farber Cancer Institute. He's also the director of the Gastrointestinal Cancer Center at Dana-Farber and the chief of the Division of Gastrointestinal Oncology at Dana-Farber. Today, we're going to hear from Brian's work about altered systemic metabolism and pancreatic cancer, early disease biology and detection. So welcome, Brian, and look forward to hearing your talk.
Excellent. Thank you, Tim. And thanks to the organizers for inviting me. So as Tim read, I thought the story I would talk about today relates to how we've been studying how metabolism at the systemic level, so the organismal level, is altered in pancreatic cancer, and in particular, how we've been trying to understand how that relates to early disease, so the biology of early cancer, and also I'll finish with some thoughts around how we might leverage this for detection efforts, so early detection. So these are my disclosures. So what was the underlying motivation to initiate some of these studies now several years ago? There really were a few. I would say one, which I think most of this audience likely knows, pancreatic cancer is the third leading cause of death from cancer in the United States. Unfortunately, the incidence is increasing, and probably in the next 2-3 years in that range, it will actually become the second leading cause of cancer death in the US. So a clear unmet need, a big problem for us to try to understand. Second is a large majority, more than 80% of patients with pancreas cancer present with advanced disease. So when they come to clinic, we already know it's going to be extremely difficult to cure those individuals, and very few of them would be cured or are cured. And so really trying to understand the biology of early disease and how we might diagnose and detect the disease earlier is really a critical aspect to how we hope to reduce mortality from this cancer type. And then the third is there are a number of features that are related to metabolism and systemic metabolism that are known to be related to pancreatic cancer, including things like obesity or diabetes being related as risk factors to pancreatic cancer. And then the fact that pancreatic cancer actually causes diabetes in some individuals in the several years before diagnosis. Now, when you sort of think about what are the cancers that are sort of the poster childs for cancer cachexia with advanced disease, where you have wasting, pancreas cancer really is at the top of that list. So it's really been associated with these different factors for some time. So really the approach we've taken is we've really tried to go around sort of this circle, which is to think about things in the clinic. I'm a medical oncologist. I see patients with GI cancers in the clinic and a lot of patients with pancreatic cancer. How do we then apply that at the population level? How do we bring that to the lab at the bench and study? And how do we continue to go back and forth between these different aspects of research and care to ultimately end up at the bottom with clinical care and actually changing clinical care to reduce mortality? So I thought I'd start with this slide, which I'll show a few times during the talk at different junctures, around clinical care. And so to the right is the yellowish orange box where there's a pancreatic cancer diagnosis. This is when I see the patient when they get diagnosed with their cancer. But in the years leading up to this, even decades before, there are quite a number of features that are related to this development of cancer. There are risk factors decades before Again, obesity and diabetes on those lists, on that list, in addition to genetics and other things. And then there are a host of things that change in the, I'd say, 6 months to 2 or 3 years, or even 4 years before patients are diagnosed with their cancer. And these are things we see when you ask the patient in clinic, what's been going on over the past few years that led to your diagnosis. These are the types of things you hear about over and over again. And as you can see, many of them seem to be related to metabolism. So elevations in glucose, changes in weight, malabsorption of nutrients, diabetes diagnoses, things that are linking this disease with altered metabolism. So we decided when we were thinking about projects and work to push into a number of years ago that we would try to understand some of this better. And you realize when you do that, you're obviously not the first person who's thought of this. And this is an article from a few years ago, as can be seen from the New England Journal back in the 1930s, from Alexander Marble, who's a physician who is in what's now the Joslin Diabetes Clinic. And what you could see is in a study that they did even back then, when they asked among individuals who had diabetes, what cancers seemed to be prominent among these individuals. And you can see in the summary statement from this article, really the one that they highlighted was pancreas cancer, that the incidence of carcinoma of the pancreas was extraordinarily high. And that really has remained true through the years. So the way we decided that we would approach this to begin with is to try to get a sense of the pre-diagnosis metabolic phenotype in patients who get pancreas cancer. We leveraged large prospective studies. So this is now to the population science aspect of the work, where we're really trying to see how do you get samples in the blood or data from patients before they're diagnosed with the disease. And this is actually relatively uncommon and difficult to get a hold of. But it turns out there are a number of large prospective studies, including several based at Harvard, where blood samples were collected and then hundreds of thousands of people were followed prospectively. And some of these individuals, as you can see here, develop cancer. Many of them don't. And you can then go back and pull samples from the freezer from years ago. Among those who had cancer or who developed cancer, really, they don't have cancer at the time, but those who develop cancer in the future versus the controls who didn't. And you can do this over many years because these studies have been going on for decades. So one of the first things we did a number of years ago was just to ask, can we characterize what has been seen in the literature in separate studies, but all in one single individual set of patients or participants? And really we wanted to know, could we see that insulin resistance was a classical risk factor for pancreas cancer, as had been seen before, meaning a decade or longer ago before, the risk of pancreas cancer is higher if you have insulin resistance or hyperglycemia. And could you also see that as you get closer to the time of diagnosis, this sort of paraneoplastic phenomenon occurs where patients can get hyperglycemia and insulin resistance in really the 2 or 3 years before their diagnosis. That's a pretty unique feature to pancreas cancer. Obviously, other diseases, other malignancies have obesity and diabetes as risk factors, but really almost no other cancer seems to have this uptick in insulin resistance and hyperglycemia in the several years before diagnosis. But in this nested set of individuals where we had blood before diagnosis, we could exactly recapitulate this U-shaped curve where we could see both of these time periods, the importance of insulin resistance and hyperglycemia. So at that point, what we decided to do was, how do we get a bit more of a broad picture, not this limited view, but a bit broader picture of what was going on in sort of the metabolic milieu of these patients before they were diagnosed, and how do we try to differentiate what was going on before diagnosis in cases versus controls. So to do this, we teamed up with Clary Klisch, who has a lab at the Broad Institute, and Clary at the time was setting up a number of mass spec-based approaches to measure circulating metabolites, or really metabolites from any fluid that you wished or cell system. And so what we did is we used that mass spec platform that Clary had been establishing. We used samples from these cohort studies where we had pre-diagnosis blood. And we asked at the time, this was about 80 metabolites, you can see here on the y-axis. We asked at the time what metabolites could predict who was going to get pancreas cancer in the future compared to those individuals who would not. And you can see a p-value of 0.05 is this dotted line. And there were a number actually of metabolites that were higher than that line, or really suggesting a p-value less than 0.05 and a statistically robust association then with development of pancreas cancer. But there really were three metabolites that stuck out the most. And these were three related amino acids, the branched chain amino acids. And we're fortunate in these cohort studies that there are quite a bit of other information available beyond just these blood values. We obviously have repeated measures of their weight, of their physical activity. We know if they have diabetes. We'd actually already measured, as I had mentioned before, markers of hyperglycemia and markers of insulin resistance. And we could really start to try to sort out how did this seem to be playing a role, meaning why and in what sort of mechanistic order did the elevation of these amino acids seem to contribute or be associated with the development of pancreas cancer. And so through a number of different analyses, trying to leverage what we already knew about being obese and diabetes and when the diabetes developed and did they develop together, really what we realized over time is that this seemed to be not dependent upon any of these other things that were already known. Right? One possibility was that we were just recapitulating what we already knew, but with a different kind of marker. But that did not seem to be true. Really, this was an independent, and it took a little while to sort this through, but this was really an independent relationship between the elevation of these amino acids in the blood and the future development of pancreas cancer. And so as we were starting to think now about taking this into the lab and trying to study in model systems how this worked, we also began to think again about time. And it's a very nice feature of these cohort studies that because the samples are drawn at a fixed point in time, but people get their disease at variable points in time after that, you actually have a large gradation in time at which the sample was drawn before the cancer diagnosis. In the same way we could think about markers of insulin resistance in this U-shaped curve, this in terms of time before diagnosis, we could also think about this in relation to the amino acid elevations that we had seen. And it turns out, as can be seen here on the sort of purple box, that the timing was quite different, right? There was not a classical risk factor, meaning if you went back 10 or 20 years, it didn't matter what your levels of branched chain amino acids were in the blood. It seemed to matter most as you got within 2 or 3 years of diagnosis, and actually even went down a little bit as you got right close to diagnosis. It turns out this is likely due, this decline due to the weight loss that we see in disease, which I'll touch on as we go. But this really suggested to us that, again, it was not recapitulating what we had seen before. And it really didn't seem like this was a classical risk promoting the disease itself. Although there have been a number of studies by other groups since this study that have suggested that may be in part true. But what we really began to think is that this seemed to be a subclinical marker of an intact tumor. And maybe the relationship was actually the reverse. And maybe there was an early tumor that was developing, and this was somehow promoting the increase in branched chain amino acids, whether it was the tumor itself or in other ways. And ultimately, this was the hypothesis we decided to bring to the lab in order to test that an early tumor formed and that branched chain amino acids rose, and that this was independent of the other metabolic changes that have been identified in patients before: obesity, diabetes, other markers of hyperglycemia and insulin resistance. And so to do this work, we teamed up with Matt van der Heijden, who's here at MIT, or here at Dana-Farber and has a lab at MIT. And so we started to leverage some of the well-known KRAS and P53 genetically engineered mouse models of pancreas cancer. And in particular, what we did to start with was really ask, could we find a time where there was an early tumor in the pancreas where we could check to see were these amino acids rising in mouse models like we were seeing in people. And those initial experiments, I show some of them here, but really showed that that was true, that at an early time point, when there was a discernible tumor in the pancreas in these genetically engineered mice, there was an elevation in the same amino acids we were seeing in people. And if we then looked more broadly and asked, well, maybe this is just a phenomenon of all amino acids, that wasn't the case in people, but maybe it could be in mice. And that really ended up, again, not to be true. That this was really quite specific for this one class of amino acids compared to others, and really suggested that this phenomenon we were seeing in people could now be studied in mouse models because we see a somewhat similar phenomenon where there's this elevation at what we think is an early time of disease. Right? Really, the benefit of this in many ways was that in people, there was not a way to go back and know whether they had an early tumor 2 years before. We might assume that they did, but we don't have imaging or other things in those studies to know that. So this allows us to investigate that mechanism more thoroughly. So then through a series of experiments, we took advantage of some of the characteristics of branched chain amino acids to try to understand where they were coming from. Meaning if they were elevated in the blood of the mice, why, right? Where did they come from to do that? And there were a few things that we were able to take advantage of. One is these are essential amino acids, so not synthesized by the body. There's really little hepatic metabolism. So what you eat gets absorbed through the intestines, passes through the liver, and ends up in the blood, and that's relatively highly correlated. So there's not a lot of first pass hepatic metabolism. And that these amino acids are predominantly stored in muscle and adipose tissue. And so through a series of experiments that Jared Mayers did, we used labeled diets with heavy carbon labeled leucine and valine, and evaluated this in tracing experiments in mice that either didn't or did have an early tumor in the pancreas, again with the same genetically engineered mouse models. And what we're able to do is systematically dissect out, was it that the intake was higher? This was leading to higher levels in the blood? No, that wasn't true. Was it that there was a difference in absorption and somehow the tumor was changing how these amino acids were absorbed into the systemic circulation? No, that was not true. We could check to see, was it that somehow the storage was slower? Mice that had pancreas cancers maintained these amino acids in the blood longer, and they weren't put into peripheral tissues as quickly, and that really wasn't true. And what turned out to be the mechanism that appeared to be operating here was that all of these aspects, intake, absorption, storage, were all the same. But in the mice that had tumors, there was mobilization of these amino acids from muscle and, well, really at this time we knew muscle, but also it turns out adipose tissue that was leading to higher levels in the blood that we could then detect, again, using the labeled diets. And so we then took from there to start to understand, well, how is this happening, right? What are some of the mechanisms behind this? And do we see this in people, right? Is this actually happening in people, or is this mostly a phenomenon that we see in mice? So to start with, it became clear looking again at these same mouse models that in the mice that had early tumors, they were not putting on or adding to their skeletal muscle and adipose tissue in the same way as mice without cancers. And you could see this both at the level of skeletal muscle and at the level of adipose as the mice developed. This is with sacrificing the mice and measuring some of the hind leg skeletal muscle and epididymal adipose tissue. You could also see this if you did CT longitudinally, so using CT to quantify volumetric measurements of muscle, again, typically in the hind leg. And you could see again that the mice that had early pancreas cancers were not adding skeletal muscle or even losing muscle, whereas this was not the case in the mice without early tumors. So this then really begged the question of whether this was happening in people. And so after some sort of searching around of how would we measure this in people and try to get a handle on whether what we see in people where they had elevations in their branched chain amino acids, do we see also this evidence of tissue catabolism? That was sort of the next step that we took. And this is efforts led by Anna and Michael Rosenthal. Michael's a radiologist at Dana-Farber and Brigham and Women's Hospital. And what Anna and Michael did is using CT scans that allowed us to then quantify adipose and muscle tissue in patients. And we didn't devise this method. This method had been around for some time and was in many ways being used to study cachexia and outcomes in patients who had advanced cancers. So basically we can use this approach to find the L3 vertebral body and essentially paint or quantify the areas of visceral adipose tissue, subcutaneous adipose tissue, and skeletal muscle. And at the L3 vertebral body, this is thought to correlate well with the total body skeletal muscle and adipose tissue in patients. And so we gathered a few colleagues and friends at a number of institutions, including in the Mayo Clinic and Mass General and Stanford and UNC. We aggregated scans and blood work and clinical data, and across about 700 patients, went through and manually essentially calculated these areas of adipose and skeletal muscle. um, and tried to ask, did these individuals at the time of their diagnosis seem to have evidence of muscle wasting? And what we saw is about 2/3 of patients with pancreatic cancer at the time of their diagnosis had what would be called a diagnosis of sarcopenia or muscle wasting. I would say that the cutoffs that define sarcopenia were not really made with this exact type of study in mind. And as I'll show later, we've been working hard to try to now understand more clearly what constitutes true muscle wasting in patients. But these were the cutoffs we had available at the time from the literature. So the first thing we assumed is that the muscle wasting that we saw would likely be in those individuals who had advanced disease, right? This is really pushing towards the idea of cancer cachexia, which is often thought of as an advanced disease phenomenon. But it turned out as we looked at this, it really was not dependent upon stage of disease. And almost the exact same percentage of about 2/3 of patients actually had sarcopenia with early stage disease. And all of these patients underwent surgery and had early stage disease. So this was not dependent upon tumor burden in this circumstance. It really seemed to be something that was happening early on. However, there's a couple caveats to this. One, again, the cutoffs are not really built for this purpose. Two, there are really no cutoffs available like this for adipose tissue. So it's hard to use these data to understand, are we also seeing adipose tissue wasting like we see in this? And then three, these are at diagnosis, right? And we're really trying to think pre-diagnosis, what's happening in the time leading up to the cancer. So this data set and this work wasn't going to answer that question. So I'll get to some more work around that in a second, but one of the next steps to this was trying to understand what was the mechanism in the mouse model systems that was causing this. And really, as we were at this point really framing this in sort of a framework of cancer cachexia, we really looked for a host of different markers. This is now work from Laura Dete, to try to find whether there was a circulating marker that seemed to be elevated in the blood at this time point that might be related to the tissue wasting. And we tested, as you can see, these are some of the ones that we looked at, a number of sort of candidate markers that have been evaluated in prior studies. And really none of these were elevated at the same time that branched chain amino acids were elevated and at the time we were seeing the tissue wasting. So none of these seem to clearly explain what we had seen. I would say though, the one that we thought maybe would be a good candidate for this and did seem to be somewhat elevated, although not at a statistically significant level, was IL-6. I just want to note that because I think this is an area we're continuing to work on, which is that in a separate study we were doing, this was a study we were doing with Pete Kraft at the Harvard School of Public Health, we were trying to build risk prediction models for pancreas cancer and really thinking, could you combine clinical factors, genetic markers, in this case, these were common genetic variants with a weighted genetic risk score and circulating markers, and use these to predict 10-year risk of pancreas cancer. And in this work, interleukin-6 actually came out as one of the markers that was elevated in the early disease time point. So I do think this requires further evaluation and study. But at the time, we really couldn't find any particular marker, at least again, taking a candidate approach, that seemed to explain this tissue wasting phenomenon that we were seeing in mice with early disease. And so Laura did an experiment that ended up pushing us in a direction to try to understand this further. And one of the things she did to try, again, to get at this idea of a secreted factor is she took a pancreatic cancer cell line derived from one of the genetically engineered mouse models and allografted it either subcutaneously or into the pancreas of mice. And from this, what she was able to see is that in the sub-Q allograft, there was not the same phenotype recapitulated with muscle and adipose tissue wasting, but it was recapitulated in the pancreatic injection model. And so the genetically engineered model with a tumor that develops in the pancreas and now an allograft model with injection of tumor cells into the pancreas could see this phenotype, but not under the skin. And this really started to suggest that there was something local going on in the pancreas that might be contributing to this. And through a series of experiments that Laura led, realized that what seemed to be going on is that the tumor in the pancreas was actually causing some exocrine deficiency in the mouse models. What was happening is there was a malabsorptive process where there was low secretion of pancreatic enzymes into the small bowel, and therefore both fecal lipids and protein were not being absorbed. And you can see in the plot below, were higher in the stool, this is now in the stool, of mice that had early tumors than those that didn't. And so starting to push away, at least for a portion of this phenotype, from a secreted factor and some of the framework that we were thinking about cancer cachexia, is starting to make us think a little bit more about local effects in the pancreas, and in particularly some local dysfunction at the exocrine level. So we then went back and tried to see in patients, is this something that has been identified? This is not our work. This is a review study that was written a number of years ago. And what it tried to do is aggregate a number of studies. And it turns out there have been quite a number of studies looking at exocrine insufficiency in patients with pancreas cancer after surgery, right? Because in that case, there are often requirements for pancreatic enzyme replacement, and that has a significant quality of life connotation for patients. And as you can see here in the post-op period, if you measure this, it's quite high, right? A large number of, or percentage of these individuals will have pancreatic insufficiency. But there were actually very few studies that had looked to see when someone first presents with localized disease before surgery, do those individuals have pancreatic insufficiency? And that's really the parallel that we thought would be most applicable to the mouse model work that we had seen. And what you can see here, again, these are not our studies, but by fecal elastase testing, upwards of half of patients with localized pancreas cancer seem to have pancreatic exocrine insufficiency at the time of their diagnosis. So again, mirroring a bit what we see in the mouse models. So at that point, we decided if we were going to study this in people, we really needed a more robust way to start to measure tissue compartments and tissue wasting. We needed to be able to do this at scale in order to understand this. We needed to also start thinking about population norms and comparing large populations of people who don't have pancreas cancer to those who do. We need to start thinking in the pre-diagnosis setting. So Michael, it turns out, is both a radiologist and also has a PhD in computer science. And so what we decided to do is working with MGH and Brigham Center for Clinical Data Science to generate an automated algorithm that would allow us to basically put a CD or pull down a scan from the clinical packs, and without any hands-on human work, spit out at the end a segmentation mask and values for the different tissue compartments. And so using a neural network approach with training and testing sets, we were able to construct such an algorithm that allowed us to put the CD, or again, pull down from the clinical packs, allow it to select the right clinical series or the right imaging series, allow it to then select the correct slice, because really the data that we have is at the L3 slice, at the CT, that slice, and then do the segmentation in an automated manner. It also then gives you the mask so that you can do quality control on that and review that manually if you wish. After doing this, what we realized is that the machine approach really was equally as accurate as the human, right? So the correlation coefficients were upwards of 0.99, and the variability by comparing a human reading of doing this when we did it manually to the machine was the same as if you had two human reads. And this was a very helpful approach for us, because the prior study, where we had been looking at around 700 cases or so, took us about 1.5 years, and we employed several radiology residents as moonlighters to help us do this manually. This now allowed us to do, as you'll see, tens of thousands of scans within a month or two. And so that's really the next step of what we did, which is how do we start to think about population scale? And how do you start to think about what cutoffs of different tissue compartments would differentiate someone who's healthy from someone who may be unhealthy, or someone who may be developing a cancer over time that, that would show you that this tissue wasting was occurring. And so this is work led by Kirti, who is a radiology resident at the Times, now doing her fellowship. And so we basically pulled down from partners, or what's now called MGB, Mass General Brigham, abdominal CT scans from over 10,000 individuals who had no cancer and had an outpatient scan in 2012 within the partner system. And what it allowed us to do is to start to think about this like growth curves in kids, or bone density, where you can start to develop z-scores that tell you over time within individuals what is normal for them at that time, and how does that normal change? Are they dropping levels or increasing levels over time for their particular age, gender, race, or other characteristics? And so what you can see here, this is skeletal muscle area among these 12,000 scans, visceral fat area, and subcutaneous fat area. And you can see the colors are by gender, so women and men, and you can see there are pretty significant differences in these things by men and women. And then also the number of individuals we had in this data set of different race, ethnicity, we were able to do this for white and for Black individuals. And you can see, again, there are clear differences by race, ethnicity. And so what this allowed us to do then is to actually construct, again, z-scores or follow-up curves that could be used to understand if someone was changing over time compared to where we would expect them to be, again, based on their age and these other factors. So this then allowed us to design a different study. And this study was really designed to get at the question of, can we recapitulate in humans what we see in mouse models before the cancer is diagnosed? Meaning when there's an early cancer, is there evidence that there are tissue compartment changes in people? And so we did this by collaborating with folks at Kaiser Permanente, Northern California, Talak and Betty. They have a very useful closed healthcare, modestly closed healthcare system that helps with this kind of work. And what we did then is use patients from Kaiser and patients from Dana-Farber and MGB to go back and look at all of the pancreatic cancer patients that we had seen. And in this case, go backward in time, right? So go backwards and try to find a CT that had been done in the years before their diagnosis. And you can see down here, the scale here is 5 years. So within 5 years of their diagnosis. Now, in these instances, the scan is done for a different reason. It's not done for pancreas cancer, but they had a scan for some other reason. And it turns out in the United States, the number of CT scans that are done is actually quite large. And about 15% of the time in patients with pancreas cancer, you can go back and find a CT in the 5 years before that they had done that could be used for such a study. And then we can also look at controls, right? So individuals who did not get pancreas cancer and were free of cancer at the time of the CT that was obtained. And we can obviously match them on a number of different features. And what this should allow us to do is to now map over time, before diagnosis, how are tissue compartments changing in individuals who ultimately develop pancreas cancer, although at the time of their CT, that's not known. It's not known they have cancer. So this is somewhat preliminary. This is the first sort of 2/3 of the cases that we've pulled for this study. So it's about 600 pancreatic cancer cases across these institutions, where we now have put these cases through the automated tissue segmentation algorithm and looked backwards in time, how do the tissue compartments change? And so time 0 here is diagnosis, and then each stepward back is before their diagnosis occurred. And if you use 3 to 5 years as sort of a benchmark, because we went back as far as 5 years, you can see there's almost a linear reduction in the skeletal muscle area as you get closer to the time of diagnosis. What we had suspected might occur based on the mouse modeling data. So as you get closer and closer to diagnosis, the skeletal muscle seems to become less and less. I would just say as again, a caveat, this is not within a single individual. This is at the population level. So you can see it's 144 cases in this interval, 143, so in this range. As we pull more scans, we are finding some individuals that actually have several scans that will allow us to do measurements within an individual over time within the same person. And then you can also see here that the adipose tissue compartments actually have a slightly different pattern. They stay relatively constant until you get close to the time of diagnosis in this 6-month period, and then there's a pretty substantial decline that we see as we get closer to the time of diagnosis. So you can't really, though, interpret these without knowing what's happening in the base population from which you're pulling them, right? So what are happening to people who don't have pancreas cancer? And is this different, or is this just the pattern that everyone has? So at this point, across the matched controls, about 1,600 patients, these are now controls without without pancreas cancer who are matched on the factors described before. And the yellow dots you can see are relatively stable, right? These people are not losing skeletal muscle area. And these are not selected for any reason, right? These are just a random selection of controls within the entire set of MGB individuals who had scans at that same time or Kaiser Permanente. And again, you can see as we look at this, a very stepwise decline in skeletal muscle area over time as you get towards the time of diagnosis. So again, really starting to look similar to what we see in mouse models where there is this steady decline of skeletal muscle as you get closer to diagnosis and then, or closer to a symptomatic tumor and then beyond. So this has really though led us to start to think about does this then relate to your ability to detect cancer early and some of the other features that we were finding. I'll talk about that in a second. So I just wanted to highlight one other mouse experiment, and then I'll shift a little bit and talk about how we started to think about incorporating some of these concepts in detection approaches. This was an approach that now has sort of formed the foundation of a number of other studies that Matt's group was doing. But it's really around the idea of how does diet affect some of these things? And in particular, in this experiment, it was around the idea that it seems some of the tissue wasting that we see is due to exocrine insufficiency, right? So what happens if you try to fix that, right? By giving the mouse enzyme replacement, so pancreatic enzymes. This is actually what we do in patients quite often who have pancreatic cancer because they often get exocrine insufficiency, particularly as the disease progresses or they have surgery. And so you can see here by giving mice pancreatic enzymes, you can rescue or increase the peripheral tissues, both adipose tissue and skeletal muscle, although it does a bit better with adipose tissue than it does with skeletal muscle. And interestingly, we had assumed that if you did this may actually be beneficial to the mouse. But really, as you can see here, among the mice who received pancreatic enzymes with a normal diet, really it didn't change how the mice did. In fact, the mice seemed to do a little bit worse than if you had just left them alone. And this has really now formed the basis of a number of experiments trying to understand how diet relates to some of these tissue compartment changes at the level of exocrine insufficiency, but now beyond that at a number of different dietary types that can correlate with different diets that are used in people. And this really becomes a question that is asked very frequently in clinic by patients. You know, how does diet affect outcome? It is a system to try to study that. So to shift gears a little bit and think a little bit about how we might use this for some detection approaches, if we go back again to clinical care and sort of how we see these individuals presenting to us in clinic, really sort of raises the point that there seems to be an interplay here between exocrine insufficiency in the pancreas and weight loss and malabsorption, and also endocrine dysfunction and hyperglycemia, diabetes. And so what we decided to do is start to try to understand how these things relate to one another in patients. So are these things that go together? Are they defining a particular phenotype in patients, or are these separate phenomenon? And so really the way that we opted to do this was again to go back to large cohort studies where individuals are followed prospectively over time. And in particular, the Nurses' Health Study and Health Professionals Follow-up Study, which are based here at Harvard. And one of the benefits of these, among quite a number of benefits of this type of approach, is that you have repeated measures of many different types of exposures. Right? And one of these can be weight. So every 2 years for decades, you actually know what someone's weight is, and they tell you based on their weight at the time when they return their information. And you also know when they got diabetes. And was that a long time before their pancreatic cancer? Again, this sort of classical risk factor or long-standing diabetes, or was it very close to the time of their pancreatic cancer? Because you get medical records and can understand timing of when diseases develop in relation to one another. And then because you know someone's weight every 2 years, you actually can assess changes, right? So you can see, does weight loss interact with hyperglycemia? And that ultimately was one of the goals of this study, was start to understand if we think some of the exocrine insufficiency is contributing to tissue wasting and presumably weight loss, how does that relate with the endocrine dysfunction we see? So this is sort of a summary slide of some of the initial data around this idea. And so if we look and see, these are risk ratios for pancreatic cancer. The exposure track here is at the bottom. So those individuals who have recent onset diabetes, most of the literature that's within 3 to 4 years of their cancer, and again is thought likely to be a direct cause of the cancer, or a direct effect of the cancer, not a cause. What you can see is that recent onset diabetes has a 3-fold increase in risk for pancreatic cancer, right? And long-standing diabetes, it's around 2-fold. This is not new information, this is known. If you look at weight loss though, and this is actually weight loss over a 2-year period at any point in adulthood, you can see that there's a gradation in your risk for pancreas cancer depending upon how much weight you've lost, right? And the more weight you lose, the higher your risk for pancreas cancer. And those of you who take care of patients in the clinic and see patients know that pancreas cancer does cause a lot of weight loss. I mean, we see this in the clinic repeatedly. And what we then asked was, well, what happens if you put these things together? Because these individually are not really high enough risk ratios to be used meaningfully for detection approaches, early detection. And what you see is that there's a stepwise and actually quite large increase in risk if these things are going together. So if you have a recent onset of hyperglycemia and you're losing weight, this really amplifies your risk, and these two factors actually interact with one another. Now, one thing I would say is this risk ratio here, about 7, is about the same as having a BRCA2 germline mutation. There aren't a lot of risk factors for pancreas cancer that have high risk ratios. There are some genetic events, and then this actually is very similar to that in a similar range. So then this made us start to think that these things seem to be related, right? That these things may actually be going together, and that the endocrine dysfunction may be accompanied by weight loss, which could potentially, and at least in the model system, seems to be due to exocrine dysfunction. And so this is a model to try to ask, well, what predicts weight loss in the time before diagnosis? And again, I'll go back 2 slides. This is really trying to predict weight change as it leads up to the cancer, meaning in those 2 years before the cancer is diagnosed, when we may have a chance to find it early, what's predicting that weight loss? And what we were suspecting based on the risk ratios that I showed you is that it seemed like recent onset diabetes was going together with weight loss, that it would actually be a predictor of individuals losing weight. And that is what turned out to be true. There were a few factors, not many, but a few that could predict your likelihood of developing weight loss before your cancer diagnosis. Older age was one. Those who had higher weight to begin with, presumably they had more weight to lose, although that could also be that there's some biology there in particular. And then those who had recent onset diabetes had a very highly significant relationship to weight loss around the time of diagnosis. And what we could see is that if you had lost 8 or more pounds, right around, again, in that 2-year period before your diagnosis, over half or approximately half of those individuals actually had recent onset diabetes that went with it. And if you liberalized a little bit the weight loss, again, there's a large portion here. So a large portion of individuals who are losing weight were also having some endocrine dysfunction and having hyperglycemia. So the next thing we started to wonder is when you're in medical school and you're in your 1st or 2nd year and you're doing your sort of history and physical course, you're always taught that if someone comes to clinic and they say they've lost weight, to ask them, did they mean to, right? Was it intentional in some way? And so it seemed like this would be a good place to try to incorporate some of that type of understanding, because presumably the cancer was causing the weight loss and that this was not an intentional weight loss that the patient was trying to do. The question is, how do you quantify that? How do you, aside from sort of being there and saying, did you or didn't you, how do you make some sort of quantification of this? And so Chen Yan, who led this project, noted that if you can measure change in weight in that time period before diagnosis, you should also be able to get at this question using data that we receive from individuals, so participants. And so what she did is the fact that these questionnaires have dietary, quantitative dietary questionnaires made by Walter Willett and others that ask about many food groups, and you can create essentially dietary indexes or health quality sort of scores related to diet. And we also ask about physical activity. So you could actually see, well, did someone seem to exercise a lot more or change their diet to be more healthy? And then that would suggest they were doing this on purpose, right? They had intentionally done things that led to their weight loss. And so essentially she put these different things together, so diet quality scores and activity scores, and created an index that allowed us to classify people into low intentionality, meaning they didn't mean to try to lose weight, medium, and high. And what we could see is that, as we had hypothesized might be true, those individuals who are not trying to lose weight, who had a low intentionality score, had a very high risk of pancreas cancer in this setting. So again, they're losing weight and they're not trying to. And that's really a marker to pay attention to. Another way to think about this that's a little more straightforward is, well, did they need to lose weight to begin with, right? So were they overweight and they might be trying to lose weight on purpose, or were they sort of a healthy weight to begin with? And again, we really see the same pattern here. If you're healthy weight to begin with, but you're still losing weight in this setting, your risk is substantially higher, right? So a substantially higher risk. And this has really led, and I don't have enough sort of data together yet to show here, but this has really led us to start to try to understand, how do you use some of these tissue compartment approaches to understand intentionality of weight loss? Are there differences in the way muscle and fat interact with each other when there's an intentionality to this, as opposed to when it's cancer related? And using some of the algorithms, the automated algorithms that we've put together, we can now do this in large numbers of individuals to start to understand this, which we hope can then provide some additional information as we start thinking about detection. So I think I'll finish with a few slides on thinking about detection and how we might operationalize some of this work. We've obviously been very interested in mechanisms and how things are working, but at the same time, at the beginning of this, really some of the idea was, if we could understand early disease, would it allow us to start to detect cancers earlier? And so this is an attempt to show you, across different sort of categories of risk, as we've been talking about, what the 4-year risk of developing pancreas cancer would be. So from that time point for the next 4 years, we use that categorization because that's the time frame in which we were looking at recent onset diabetes. So diabetes and then the 4 years after. So you can see, if you look at all participants in these studies, across these cohort studies, the 4-year risk is very low, right? So 0.09%. If you have recent onset diabetes, your risk over the next 4 years goes up. You're at about 0.4%. And then as you add each of these things, weight loss, lack of intentionality of weight loss, or say now an older age and lack of intentionality of weight loss, you can see how the risk over this time period increases. So to 1.5% or 2%. Now you're in a range where you have 1 in 75 or 1 in 50 people will actually get pancreas cancer in the next 4 years. I would say that these risk estimates are based on the populations that we looked at, right? These 2 large cohort studies. They're clearly applicable to the larger population, but there obviously can be some differences between this population and the general US population. So what we started to think about from here is, are there ways that you can start to use blood-based approaches to then enhance your ability to detect cancers among individuals with risk in these ranges? And so I'll just very briefly talk about this paper, as I see I'm getting a little lower on time. So this is a manuscript that we wrote together with Sam Hanash's group at MD Anderson, and really trying to characterize a marker that is well known, right? So CA19-9, it's a marker we use in the clinic all the time to follow patients. It's really not used, though, to detect cancer early in any large degree yet. And so essentially we put together across 3 centers, Dana-Farber, Brigham, Beth Israel, and Columbia, an early stage cohort. So these are all patients who had very early stage pancreas cancer, went to surgery, about a third of them had stage 1 disease, and then a set of controls, and asked what's the ability to discriminate cases versus controls using CA19-9 for early stage pancreas cancer. And what we could see is that the area under the curve for this is an ROC curve, is about 0.9. And if you choose a high specificity to limit false positives, it's about 65% sensitive, okay? We then added to this samples that Sam had acquired from a different prospective cohort, the PLCO cohort, but with the exact same design as what we showed at the beginning, where we were looking at metabolic alterations in branched chain amino acids. And what you could see here, this is a time-based approach, again, similar to what was described before. The y-axis is sensitivity at high specificity. That in the 6 months before diagnosis, it's about 60% sensitive and 99% specificity. So relatively similar actually to an early stage cohort at diagnosis. But there is a significant drop off as you go backward in time. And again, this is going before diagnosis. Now it's about 24% sensitivity in the 6 to 12 months, and then it goes down from there. So it really suggests that some of these tumor secreted factors, and it's not clear exactly how generalizable this is to the many other things that people are studying, but tumor secreted factors, this may require that we measure blood samples more frequently if we're going to use this in a screening context, whereas some of the metabolic changes we described before seem to occur earlier because they were actually, we think, related to the whole body reaction to the tumor, not just the tumor itself. All right, and then I'll finish up with a couple slides. Just if you sort of take this to the next step and sort of carry this forward and what this would actually mean. So if we focus on those individuals where we have 3 factors present. So diabetes that occurred recently, they lost weight and they didn't mean to, so they had a low intentionality score. Again, this is using the same cohorts we described before. And we have a test that's like CA19-9, 65% sensitivity, 99% specificity. You can see the positive predictive value goes up as the 4-year risk goes up to about 2/3 in this situation. The negative predictive value is quite high because most of the individuals don't have cancer in these groups. And the number of cases you would need, or number of individuals, not really cases, the number of individuals you would need to screen to find one case is around 100, a little over 100. And you can sort of play that out down here and say, well, if we were to screen this 106 individuals, there would be 2 cancers on average within this group. One we would find, one we wouldn't, right? So one would be a true positive, one would be a false negative, because the blood test is not pure, it's not 100% sensitive. And most individuals would not have cancer, and those would be true negatives. And then there would be one false positive, right, from the test, where we would tell someone, well, maybe you have cancer, but they actually didn't, right? And we'd have to follow that up. So you can start to, as you start to have absolute risks from defined populations and sensitivity specificity metrics from defined blood tests, we can start to think, if you operationalize this in a population, what it would mean. And so to sort of finish, I would say this has now really led us to think more about all these different factors that are occurring in the time before diagnosis, with really a particular interest in this several years. And I think we've focused in the discussion today on sort of endocrine dysfunction and exocrine dysfunction and weight loss and diabetes, but there are many other factors that seem to repeatedly come up when you see patients with this disease in the years before. And so a big project that we now have ongoing is part of a team that's funded by the Lustgarten Foundation and Stand Up to Cancer, is how do we start to think more broadly, be less hypothesis based on only these specific factors, and start to think, how do we use real world medical records more efficiently to start to construct models that would allow us to understand risk and apply some of these metrics. And so this is ongoing work that I don't really have time to talk about today. So I'll finish there. Obviously a huge team effort to do this work, and a lot of thank yous, including Anna and Chen, who did a lot of the work in the human studies. This has been a long running collaboration with Matt Vanderheiden in his lab. We obviously have benefited greatly from Clary Klish's expertise and working with him now for many years. Michael Rosenthal, who really led a lot of the radiologic studies that were shown. And then this work included collaborations with MD Anderson and Kaiser Permanente, Mass General, the Harvard cohorts, and the Mayo Clinic, and then obviously the funders of the work. So thanks for everyone's attention. And again, thank you to the organizers for the invitation.
Thank you very much, Brian. That was a fantastic talk, very interesting. There are a couple questions. Let me take the chair's prerogative, though, and ask you one first. The data that you showed suggested that something is happening, something potentially interesting is happening in the couple of years right before diagnosis. And I know, you could talk a little bit about the potentials for, I don't know if bias is the right word, but other things that might explain those. But my question is really, do those data that you presented give us any information that might reflect the latency of disease or the disease progression? Is it telling us that there's really nothing biologically tumor-wise going on before a couple of years before these diagnoses, and then it picks up really fast and takes off? Or is there anything we can make of that about the period right before diagnosis?
Yeah, thanks, Tim. I think that's a fantastic question. I'm not sure we could say from these data with clear specificity exactly when there's detectable biology. I think there most likely is biology going on before this period, but we have not, in the studies that we've done, detected it prior to that time. Now, I think there's a lot of debate in the community of people who study pancreatic cancer, what the trajectories of this disease are. And there are sort of a couple hypotheses on sort of slow accumulation of genetic events that happen over long periods of time. There's also thoughts around more catastrophic events where there's chromosomal interruption and it's a more rapid disease formation. I would say my sense, and this is informed in part by these studies, in part by being in clinic, there seems to be both. We do see patients where they come to clinic, they have a pancreas cancer, we review their scan in tumor board, and 9 months before they had a scan for an unrelated reason, and we can actually see the cancer there. It's just it wasn't seen at the time. But there actually are also quite a number of patients where the reverse is true, where they present with metastatic disease, they had a scan 9 months ago and there was nothing there. And I think there's really going to be divergent biology. I don't think this is all the same. And I think there are a number of studies, including some recent work by Dave Tuvison looking at different injection mouse model approaches using organoids, showing that there are divergent biology. And some of these really do seem to take off. And those make me nervous because I don't know how we're going to find those ones early. I had a patient recently I saw where he had a scan 7 months before, there was nothing, and he had metastatic disease when he came to see me. That's just a scary thing. But there also is the reverse. And I think there's a lot more work to do to understand the biology of what's determining this difference in sort of rapidity of growth at that time period.
Yeah, great. And something that'll be really important if there's ways to understand heterogeneity of those progressions or tumor subtypes or whatever. Great. So a couple of other questions. Earlier in the talk, you were talking about sarcopenia. And one question is sarcopenia specific to pancreas, or is it KRAS P53 driven, therefore will be true for lung cancers, at least in mouse models?
Yeah, that's a great question. So in one of the first papers we published, we looked at this question. We looked at it less from the sarcopenia standpoint and a bit more from the elevation in branched chain amino acids, because which we think now is related to the sarcopenia. But we were interested to see, was this specific phenomenon to pancreas cancer, or were in other mouse models, could you see this? And so using KRAS P53 driven alleles in both lung and for sarcoma, we did not see the same elevations. So in mouse models, we did not see this. There now have been a number of groups that have done similar analyses in patients, in humans, where we also don't see this. So I think sarcopenia is clearly a more broad phenomenon than just in pancreas cancer. But there is something different here. And I think some of that may relate to some of the exocrine insufficiency phenotype here that we've seen in the mouse models. There's no reason why other diseases, other cancers should have that. This is a local phenomenon in the pancreas, but there may be other biologic differences too that are defining that. And I think one of the things we're doing now is we have a large project actually to understand weight change and intentionality across all cancer types. Just sort of drove our interest to figure that out. And this is preliminary, but across a host of different cancers, we don't see weight loss: breast cancer, ovarian cancer, hematologic malignancies, but there are a subset that do. Pancreas cancer is sort of way at the top of that list, but there are a few others that do also. And I think ultimately there may be some disease-specific biology we're going to have to understand. And I don't think cancer cachexia is just one thing. I think this phenotype is going to have to be understood at the level of each disease.
Great, thanks. Let's take a couple of more questions. Stephanie Price asks about coming back to the intentionality of weight loss. Could it be that the diagnosis of diabetes and the following change of diet induces weight loss even without intention? And so the patients may also lose weight by thinking more about their diet or, yeah.
Yeah. So I think that's true. I mean, I think we were wondering whether the weight loss you see among those people who got recent diabetes was just because they were eating healthier, that their doctor was telling them to do that, which is sort of the standard counseling you get if you get a new diagnosis of diabetes. But the truth is that didn't really bear out, right? Again, really, as we assessed this intentionality, it's really not asking anything about whether you changed what you were doing because of a prior diagnosis. It's just objectively asking you, what did you do?
Mhm.
And so you could see that even among individuals who had recent onset diabetes, there was unintentional weight loss in that group. And that really went together with the weight loss we were seeing. That being said, I think there's a lot more to understand there. And I think we really need to dive into that more deeply, because I do think that could be useful to find, so you don't have to ask someone, what are some objective measures of when this lack of intentionality is occurring? And how do you then leverage that to understand, oh, I should be worried about cancer in this person, but not the next person?
Yeah, got it. One more question. The TGF beta often tracks with cachexia. Have you measured this with your branched chain amino acids? And can TGF beta be another marker for pre-pancreatic cancer?
hmm Yeah, I think it's also a great question. We haven't measured that specifically, no. So I can't address that question directly. There's been, and it's not our work, but there's been further work really showing that there is a lot of interaction between stromal cells in the pancreas and tumor cells, and that there seems to be a sharing of branched chain amino acids between the cell types, which may be important to drive cancer growth. And some of that seems to be mediated by TGF beta. I think it's a great point and a great idea to look at that more in depth. We have not done that so far, but it really does, I think, speak to an important area to consider.
Great. So in the orthotopic implantation experiments, did you also test non-pancreatic cancer cells?
Mhm. That's a good question. So not into the pancreas,
Mhm.
into other organs, yes, where we did not see this phenomenon. Um, I'm wondering, though, whether the question is asking, do we inject them into the pancreas, which is a very interesting question. I think back if there's anything we did that would get at that. Uh, Not really. I don't recall us doing something where we injected other tumor types into the pancreas to look at that question.
Mhm.
But I think it really does, it's a good point, because part of this you wonder, is it just a physical disruption, right? That there's actual obstruction of ducts in a physical manner
Mhm.
that then leads to reduced secretion of enzymes into the bowel? Or is there actually a biologic relationship between some of the acinar cells that are making the enzymes and the tumor?
Yeah.
I think our hunch is it's a bit of both. We tried to get at that question a little bit by asking, where was the tumor in the pancreas? You might suspect you would have worsening of your tissue wasting if it was a head tumor that blocked more of the pancreatic duct and therefore didn't allow enzymes to exit, versus a tail tumor, which would do less of that. We did not actually see much difference in that circumstance, meaning there seemed to be a pretty similar degree of tissue wasting, whether the tumor was in the tail or the head. That being said, that's a pretty rough, way to look at this. And I think that requires a bit more investigation to look at that further. But I think that's a great point, that the more we understand about how this is working, the better we can try to, address it in patients.
Great. We have a couple more minutes. Let me just ask a couple more questions. It looks like the diabetes that are good indications for PDAC are type 1 diabetes rather than type 2. Is that right? And could you tell from your data how likely it is that a person who recently suffers from type 1 diabetes will likely develop PDAC in a few years?
Uh-huh. So I would say this is really predominantly type 2, right? So type 1 being sort of young child onset, lack of insulin secretion. That's really not this circumstance. And I must not have made clear, these cohorts are all adults. So these are people getting diabetes in their 50s, 60s, 70s, right? So this is almost exclusively type 2 diabetes. I do agree that it seems that the particularly high risk is in the recent time period, right? It is also clear, though, when you look at the data, if you have diabetes now and you're 50, and you follow all those people with diabetes for 20 years, those people with diabetes will have a higher risk of pancreas cancer 20 years later than controls who don't have diabetes. So there is a risk factor component to this. And it seems across the studies, it's about twofold, right? But if you look at that specific time period right after the diabetes is diagnosed, the risk of pancreas cancer is substantially higher during that interval. And that really, again, seems to be this interplay between the tumor and cells in the pancreas, and potentially peripheral tissues too, that leads to this insulin resistance, similar to how it looks like that tumor there also leads to the exocrine insufficiency and presumably some of the malabsorption phenotype. So these things are going together in that pre-diagnosis period in sort of that 2 or 3 years.
Great. Well, we're at time, and so I just want to thank you. There are a couple more questions that we'll send to you to respond to if you can, but this was very fascinating, really important work, and I appreciate you taking the time to speak with us.
Excellent. Thanks, Tim. Thanks, everyone.